A lot of students understand how the Lac Operon works until the AP Bio exam. Hi, I'm Miss Parrott, and in this video, we are going to be looking at how the Lac Operon actually shows up on the AP Bio exam and the kinds of questions that you're going to see on the test. So this is a quick picture of the Lac Operon itself. If you haven't already, check out the structure, the Lac Operon structure video. And because we're going to jump right into this, assuming that you understand what all the parts are and how they work and what they do. The test is going to not just see, do you know the parts or what, like the functions of each part. It's going to ask you to apply your content knowledge in a test scenario. But what's great is that fundamentally, it's really just asking one question. Given these different circumstances, will transcription of these structural genes occur? Remember, this whole first part, the cap site, the promoter, and the operon, is all part of what either turns this off or on. And then also the cap site then is going to help us regulate, are we on low transcription or high transcription? So when we think about will transcription occur, one of the main contexts they put it in is sugar presence or absence. Now remember, lactose is the sugar in milk, and this is happening in the E. coli bacteria that live in your guts. So when lactose is present and glucose is not, then your body's going to want to cut this so that, it can make glucose, which is E. coli's favorite fuel source. So when lactose is present, yes, transcription will occur. And when it's absent, no, it will not, because the bacteria are trying to save energy. That's really the whole point of having operons in the first place. Glucose's role, again, it is the gas in the gas tank. That's what the bacteria wants to use for energy. If glucose is already present, then and lactose is also present, transcription will occur. It'll just be low. And if glucose is absent and lactose is the only thing there, then it will crank up the transcription of these genes to high so that we can convert lactose to glucose. Because we need it, because there's no other source of energy. Then when lactose goes away, the repressor goes back on, and we're good. So, how's it showing up on the exam? Now, it could be about lactose presence or not. It could also be about the proteins that are involved in the transcription. So remember, RNA polymerase attaches to the promoter and will move down this strand of DNA and transcribe LAC-Z, LAC-Y, and LAC-A, which are all needed to process lactose into glucose. If the repressor is active, then will transcription occur? No, because the repressor is blocking RNA polymerase. If the repressor is not there, hey, then yes, we will get transcription. I encourage you to make yourself a little set of these things and play with it so that you can kind of play out different scenarios, just as I'm doing. Now remember, the cap protein and the cap site is activated. The cap protein is activated when cyclic AMP is high. Cyclic AMP is high when glucose is low. And so, when, again, glucose is low, that's our gas in the gas tank. So we want to be able to turn lactose into glucose. So cap protein is going to come here with its friend cyclic AMP and say, RNA polymerase, over here, come join me. We need your help. And so RNA polymerase, it helps it bind to the promoter so that then it can transcribe our our genes. The genes will form, or the transcript will be translated into a protein, and the protein will cut the lactose into glucose. And now we have more gas in the gas tank, right? That's the whole idea. So your question may be asking about the repressor and its activity, and then the cap and its activity. So if the cap is active, you're going to have high operon action and transcription. If glucose is high, then cyclic AMP is low, meaning your cap protein isn't the right shape to connect to the cap site, so it can't do it very well. So RNA polymerase will still attach, but not as well, and so you'll have a low rate of transcription. So with those fundamental things in mind, what kind of question am I going to see on the exam? That's what we're looking at first. All right, so for first, the multiple choice. Now, AP Bio loves to make things way more complicated than they need to be. So if you're not already strong with reading, start reading old AP Bio questions, because understanding the content, unfortunately, is not enough to be making like a 4 or 5 on the exam. So the first type of question is going to ask you about sugar conditions, like we saw in the first tree. So lactose, is it present or absent? Glucose, is it present or absent? It's going to hide that information in either a very wordy scenario, and then like in the last couple lines, it'll be like, which of these is true if lactose is not present, right? Like it'll slide it in there after 2 paragraphs of extra information. You may also find this hidden in an illustration. So in some past questions, they'll give you like a wordy scenario, and the answer choices are actually pictures of the operon, like some with the repressor on, some with the repressor off, some with at lactose there, some with lactose not. And there's a key, and it seems like a lot, but it's really not. So expect that. Again, all this is about going in, knowing that we're going to see hard things, knowing that we can do hard things because we will have practiced them, and then actually doing the hard thing, and then scoring well on the exam. Hey. So it may also hide it in a data table. It may give you something like this and then ask you a question about it. So, but again, we just went through the different sugar conditions and how they're going to impact whether it's on or off, and then whether it's high or low based on your glucose. All right. The second type is the what if mutation style question. AP Bio loves this with all kinds of anything that's some big involved process. So actually what I'm going to walk through here, you can apply to like all the proteins or the steps that are involved in photosynthesis or in cellular respiration or in, meiosis. Like if these things go wrong, if step 1 goes wrong, what happens to the rest of the process? If step 2 goes wrong, what happens to the rest of the process? That is a great way to study. So let's look specifically at the Lac Operon. Now, there is a gene called LAC-I. LAC-I is not in the Lac Operon. It actually is a gene that's farther like upstream of these on the prokaryotic circular DNA, but it codes for the inhibitor or the repressor protein. So if there's a mutation in the gene that codes for the repressor, you're going to have a mutated repressor. And if you have a mutated repressor, then there's nothing stopping transcription from happening. So your operon is on, and yes, transcription is happening. If we move down the line, there is a mutation in either the gene that codes for the cap protein or the cap site itself, then RNA polymerase is not going to be able to bind as well. And so It's kind of like if instead of having a giant flag saying, hey, come over here, you just have like a tiny flag, or your flag is like on the ground. Or you don't even have a flagpole if the cap site is messed up, right? So RNA polymerase won't bind as well, so you may have transcription if lactose is present, but it'll be low. If you have a mutation in either the promoter region of the operon or the gene that codes for RNA polymerase, then you're gonna have a mutated RNA polymerase, or the promoter won't be bindable, it won't be able to latch on. And so if RNA polymerase can't bind, its job is to transcribe. So if it's like a train that can't get on the train tracks, then it's not going down the road, right? So no, it's going to be off. Now, what if there's a mutation in the operator? The operator, just like the promoter, is a non-coding sequence, but its structure is important for the repressor protein to latch onto. So if we have if a repressor protein can't latch on, then it can't stop transcription from happening, so transcription is gonna go. Now finally, what about the structural genes, the LAC-Z, LAC-Y, and LAC-A genes? Well, these genes are not involved in the turning the operon off or on. Those are not regulatory genes. Those are the structural genes that will make the proteins that are in charge of turning lactose into glucose. So structural genes, since they don't regulate gene expression, then you'll still get transcription. However, depending on what is impacted, where the mutation is happening, you may have an impact on how well, if at all, your lactose is going to be able to be converted into glucose. But it's not due to lack of transcription. So, next. These types of questions, graph and data questions, can be found in FRQs or in multiple choice questions. So, you may see a graph like this, where you see glucose level low or high, or it could be lactose level. It depends on what it is. And then what's the resulting beta-galactosidase activity? Now, you don't have to know that LAC-Z codes for beta-galactosidase. You don't have to know that beta-galactosidase is the enzyme scissors that actually come through here and cut and turn lactose into glucose, but that's what it is. Everything, you should be able to answer the question based on the graph alone. But it does help you to know how the operon works, because it could ask you so it could say which of these best explains the results of this experiment, right? And then you would say something about when glucose is low, cap is able to attach to the cap site because cyclic AMP is present. It can attach to the cap site, and you're going to have greater transcription and production of beta-galactosidase. Now, you may also see something like this. So here, this is a question. It gives you 4 different conditions or scenarios, right? And lactose, glucose, and then enzyme activity. So minus means absent and plus means present. And then enzyme activity as very low, low, or high. And then it would ask something like, which of these conditions shows positive regulation by cap? Positive regulation means is turning it on. So cap, we know, has to do with glucose. So it's when glucose is low, so that's going to be minus. And lactose does have to be present for our operon to be on. Then we're going to get high enzyme activity or enzyme transcription, and then hopefully activity. So These questions take a little bit more thinking, but if you can anticipate not just seeing the scenario described in words or pictures, but as a graph or as a data table, then you can go in when you see a question like this and not be shocked, freaked out, and think you don't know. You can be calm, cool, and apply your knowledge of the Lac Operon, thinking, will transcription occur? And if so, what are the results? All right. Now, FRQs. This is kind of a lot. So feel free to pause, zoom in, be sure you're in full screen, okay? Now, there are 6 different types of FRQs. I have a whole video on the 6 different types of FRQs. Check it out. Any topic could show up in any of these 6 ways. So there is the experimental design question, which is very question 1. All right. And so it could say something like, design an experiment. Now, what's kind of good is what you're going to have to do really in a question like that. It may use the Lac Operon as context, but you don't have to have a ton of deep biological knowledge on it to be able to identify the variables and describe what's being measured and how, and like controls and things like that. You may see data analysis. So a data analysis question, that'll usually have a graph or a table provided, and then you have to either explain why you're seeing the trends that you're seeing. You have to describe and explain. That's usually it, like the trends that you're seeing. There may be a concept explanation, which is very like FRQ number 4. And it's going to ask you to explain how, and this is when our what if something goes wrong, that's very question 4 FRQ. So explain how glucose impacts the activity of the Lac Operon, right? Or how a mutation in the LAC-I gene, which codes for the repressor, impacts transcription in the Lac Operon. So here you got to show cause and effect. And this is very like, what if something goes wrong? So studying in with that question in mind is actually a great way to study. You may see a model or a diagram. They may give you one. They're not going to ask you to draw the Lac Operon. Okay, but they may give you a model, and that of it, it could be when it's on or when it's off, or when the cap protein is there or when it's not there, or when lactose is present and absent and glucose present and absent. You don't necessarily know, but it may describe a mutation or again have the labeled operon, and your job is going to be predict the outcome if this is the setup. So they're going to give you a visual, and then you have to connect the dots in the visual to predict what's going to happen next. Then there are quantitative, there's a quantitative question. So you may, in this, you're going to be seeing like numbers and ratios. So instead of just like present and absent, it may give you specific concentrations. You don't have to know like what counts as high lactose concentration. Just look at like, compare the numbers to each other. Is this one greater or less or 0? And like work from there. And your job is going to be to interpret. So you may have to do some calculations of some type. But really the points come from you being able to interpret what those values mean in the biological context. And then finally, the final FRQ type, it's gonna ask you to predict, okay? So in this, it may give you a scenario or a table with data like you've seen before. And your job is just gonna be to determine, is transcription gonna happen or not based on the scenario, and then to justify your answer using data from the scenario, which again, may be in writing, it may be a graph, it may be a data table. So the more practice you get with those kinds of things, the better. Now, I realize that I know the Lac Operon pretty well. It took me a while to get there, okay? So if you're not fluent yet and you couldn't answer all those questions quickly, efficiently, and with time pressure, that's just not yet. Right now, it's the end of March. You have a month and a half until the AP exam. So I gave you several study tips, but what you want to be thinking to go from no to can do to fluency, so you know you're at this, I'm like ready to roll with it, if you can predict transcription instantly. So in my little tree branch, my little will transcription occur given lactose or glucose, and if the repressor or the cap protein is present or absent, like you need to be able to answer those instantly. And when you can, you know you're moving into this fluency mode. If not, that's okay. It just means you need more practice. Also, can you explain why a cause will result in a certain effect in writing with your hand, not just with your mouth? You may want to start there, but then can you explain it in writing in a way that someone else will understand? And can you interpret data from tables like the ones I showed you or graphs? Can you take those pictures of information and turn them into words, or can you take words like a long scenario describing, some Lac Operon environment, and like turn it into a picture and demonstrate what it's saying? If the answer to those things is not yet, that's okay. You have 6 weeks. We are going to get there together, all right? So remember, practice builds speed, and AP success isn't built on memorizing facts. It's built on pattern recognition. So if you can start to recognize these patterns and become fluent in these patterns, then no matter what the exam throws at you, you're going to be a success. If you found this video helpful, please do give it a like. Subscribe to my channel for more AP Bio exam skills videos. I also have lots on like how all the formula and equation sheet. I have unit videos. So check all those out. Leave a comment, a question, or an emoji. And if you're a teacher, please do check out my resources at Teachers Pay Teachers at the store Know and Grow with Gnomes. Keep learning, keep growing, and we'll see you next time.